Hi, my name is Margot, and I'm a graduate assistant working with the Biological Sciences Department to support writing in the research courses this year. And I'm very excited to welcome you to our second writing workshop focusing on writing the methods and results sections of your scientific research articles. Just a reminder, these papers are due by 5 p.m. on December 10th, and to please contact me if you would like to schedule a writing conference anytime before December 1st. I'm happy to help you with your writing. So let's get started. Let's bake banana bread. So I love baking and I love banana bread. So when I look up a new recipe, I'll do a search and all these results come up. So how do I choose? Well, I usually go by the picture. So to me, this banana bread looks incredible and I want to make this loaf exactly. But could I do that given this recipe? Well, the answer is no, we couldn't. The steps are all out of order. Well, we're missing step 5 entirely. The ingredients are vague or missing. We don't know the quantities. We also don't have any details. We don't know the temperature, the time or duration of baking, or if there's any special equipment involved. So no, we couldn't make this banana bread. This is what I'm looking for. I'm looking for a recipe that will give me both the ingredients and the instructions so I can follow each step exactly and be able to make this loaf of banana bread. So very similar to baking, when you are writing the methods section of your research papers, you should provide enough detail that somebody could replicate your steps and be able to do your experiment all over again. So the goal of the methods section is to describe how you approached your research question and obtained the data. Provide clarity and detail so that your methods could be replicated. It's important to be as transparent as possible when describing your methods. This is because the readers need to know exactly what you did so they can accurately assess your results and conclusion. This again ensures the rigor and reproducibility of your study. So let's start out with a breakdown of the guidelines. The method section is organized in this way and begins with using the past tense. So when you write your method section, you've already done the research, so use that past tense. In our last discussion, we talked about using the active voice because it's really helpful in writing because it allows you to be concise. But in the method section, you're actually interested in emphasizing the action and the object. So it's okay to use the passive voice here. In the method section, you also want to use a narrative structure. So what this means is that unlike a protocol you might follow in your lab where each step is enumerated, you're actually going to write about each step you did as a paragraph. So one thing you can think about is that you're journaling your experience of doing the study. So again, avoid directives and enumerated steps. Describe what you did. So here in this example, instead of writing place the sample under the microscope as a directive, say the sample was placed under the microscope or we place a sample under the microscope. Either one is acceptable. It's okay to say we. Additionally, you're going to want to report these steps in chronological order, starting at the beginning of your study. What was the first experiment you did? And this is because some of experiments might build on each other. So it's important to clarify that for the reader. You'll also want to use subsections in the method section. So when I first started writing these papers, I often felt pretty confused because how was I supposed to write with flow to connect these seemingly disparate methods or experiments? So one thing that really helps me was using subheaders. So think about it in terms of a project being a collection of different experiments or analyses. So for example, in my project, I have a field component, lab, and bioinformatics components. So each subheader will delineate one of those components, and I will go into detail describing what I did for each. This really helps the reader kind of follow what you did and also kind of understand all the details that went into each component. You want to provide detail on your experimental design. Again, this is to replicate it. So you want to, again, provide that recipe for replication so somebody could follow your steps and do what you did. So to do that, we need to be very specific. So you need to report the materials that were used to conduct the study. So what buffer did you use? And be specific enough where you actually state the chemical composition. What gases, chemicals, reagents, equipment did you use? If you did a poll, what was the questionnaire? So in addition to describing what equipment you used, also describe the settings. So for example, if you vortexed a sample, did you vortex vigorously or lightly? It's important to make this distinction because it could have a significant impact on the outcome. So next, you want to make sure that you're describing your experimental conditions clearly. So you want to state the climate, the pH, the doses, how many replicates you used, describe the study subjects. You want to, again, include this important detail so somebody could actually follow those steps and come up with similar results that you did. You also want to include the positive and negative controls. And then if you use study subjects, you want to describe those study subjects. Also include how your organism was obtained and maintained. Again, this is all in the interest of replication. It's important to be able to redo studies to determine if you actually do get the same results or something different. That is important information. So one thing to do in your writing, if you find yourself really struggling to describe this complicated experimental setup, you could also include diagrams. So this could, again, provide some sort of visual representation of your experiment for the reader, and you would provide that information in the methods. So in addition to providing each step, you need to explain why you did each step. Why did you choose a specific design? This is incredibly important when you are discussing a novel protocol. So this might be something that you and your lab came up with that hasn't been done before. And when you're describing this, you need to provide additional context so the reader knows why you chose to design it that way. So in this example, if you capped a test tube immediately after adding a solute to a solvent, why did you do that? Well, you could come up with something like this to explain that. Well, we added solute X to solvent Y and immediately capped the test tubes. You explain what you did. That's the step. Now you provide the context. Capping the test tube ensured nothing would leak because solvent Y evaporates quickly. So that's the rationale. You explain your thinking to potential critics that way. And you make it very clear and, again, transparent to your readers why you chose to design your methods this way. If you're using an established protocol, it's a little different. You don't have to provide quite as much justification because it's already there. So if you're referring to a specific paper where they describe the methods they used and you're using those same methods, just cite that paper. For example, if you are extracting DNA using a QIAGEN kit, then you could simply state that you're using that kit. And then the reader could then refer to the manual of the QIAGEN DNA extraction kit to get the directives they need. It is important, though, if you're using an established protocol, to state any alterations you might make. That's because they're unexpected. So for example, if we wanted to remove all the salts from our RNA, we could add, to ensure purity of our RNA, we added a second ethanol wash step. And again, this explains very clearly what we did and why we did it. In this example from Lower and Hall, they're talking about extracting genomic DNA from fireflies. So they're specifying what they did. They extracted DNA from the thorax. So here they're being really clear about what tissue they extracted DNA from. And the reason that's important is because, again, if someone were to replicate this, well, they might get a higher DNA concentration by extracting DNA from the thorax as opposed to a leg tissue. So by clarifying that, they're making it easier for someone to replicate this work. Additionally, they're stating what established protocol they used, a standard phenylchloroform isoamyl alcohol protocol, and they're adding this extra step, this RNase digestion. So that's all out on the table, very clear and obvious what they did, and they're even providing this reference. Another strategy that you can use to organize your method section is to use to do X, we did Y format. So I really like this format because it's very helpful to present your objective clearly and then state your approach. Like, why did you do that? What did you do and why? And so you'll usually find this format at the beginning of each paragraph, and you'll see it in both the methods and results section. So let's look at these examples from Landers et al. 2013. So here, from the subtitle, we know we're about to read about how these researchers analyze their data. Now, to break down the to do X, we did Y format, we can see that their objective is to study the effect of background on spider color change. Now we can see the approach they took to answer that objective. Let's look at this example. The subheader now states, effect of molting on spider color change in the laboratory. So that is the title of this, or the topic of this paragraph. They're describing the experiment they did to answer or address this question. So you can see in the to do X, we did Y format, they're stating to determine the effect of molting on color change from yellow to white, again, their objective, and then their approach. We used 80 field collected spiders that were initially yellow. Very clear. And so I'd also like to add that, again, this structure is used at the beginning of a paragraph, and then you'll usually follow up with additional details describing the analysis you did, again, being as specific as possible. So let's delve into an excerpt from the Methods of Lower and Hall, 2018. And so let's look at how these authors use different components of the methods section in their writing. So first, they started out with a subheader. So this section will describe how they obtained their samples and how they extracted DNA. So we can also see that they're giving us information on how they collected fireflies from natural populations. So you can see the specific species they're interested in, P. pyralis. You can see that they collected P. pyralis from 12 different natural populations. They're even defining population as location. And then they're giving you information on the time frame. Now they're describing what variables they measured in the field. That's important because this is how they collected their data, what they were interested in. Then they stated up here that they were interested in this P. pyralis species, but there are actually many different types of fireflies. So it's important that they're recognizing the right one. They don't want to collect the wrong type of firefly if they're focused on P. pyralis. So here they're explaining to the readers how they went about that. So they watched the firefly flash pattern and also looked at their morphology to confirm that it's a P. pyralis. But as you read further in the text, you'll see that they did an additional confirmation. So in this case, they actually sequenced part of the CO1 gene, and then they compared it to an in-house database to confirm that they're actually looking at a P. pyralis specimen. So this is a really great way to ensure the rigor and reproducibility of your data by, again, being transparent about what you're doing and then including these different checks. So to kind of show to the readers, this is a legitimate way to confirm species identification. They're actually stating here, this mitochondrial locus has been shown to be phylogenetically informative in Photinus fireflies. And then they're providing that reference so the readers can look it up. So that's really important context to provide to explain their rationale for doing two different species identifications. So normally this would be an interactive activity, but I will walk you through it. So we read this excerpt to ourselves and tried to identify where we could find this information. So using the to do X, we did Y format, we're able to find their objective and approach. So we know what we're about to read, that they did a really great job of framing that. So now we can kind of get an idea of the sample size, what data they collected. So you can see that all right here. The final data set included 191 individuals, and they sampled 15 to 16 P. pyralis from each population. And they even summarized the color of their light. So this is all important information, kind of again, summarizing what they collected. And so then if a reader that doesn't know much about fireflies is reading this, they might notice, well, why did you only use male measurements? And so to kind of address that question immediately, these authors are then kind of explaining, well, female fireflies are actually really hard to find in the field. They perch on these leaves. And so it's a lot easier to find males. So probably in the interest of achieving a high sample size, they focused on using male measurements. And so again, providing that justification very clearly is helpful to be as transparent as possible for the reader. So one thing that can be very difficult when kind of summarizing your research and identifying, well, what are the key steps? What should I actually be focusing on in the methods? Because you don't need to state everything. So this kind of series of steps is inspired by a Biology 1108 lab. And so each step might be something that a student would do while conducting their experiment. And so imagine they wrote this down in their lab notebook and then are interested in writing up a report, like the methods section. Well, what steps would they want to focus on? So we looked over these steps and then we tried to identify what the key steps were. And so I'll walk you through what I consider to be the key steps. And I will also mention that you, as the scientist behind this study, are the expert. So you will know what steps were most important when designing your study. So think about those. What were the most important steps in getting from point A to point B that you did in your study that you should then emphasize? So with that being said, these were the steps that I thought were important. And so you'll notice that a lot of steps that may seem important, like recording observations, are not highlighted. And that's because I would argue that that would be implied. So as a reader, I'm assuming that the authors of these studies have done rigorous research. So in that assumption, that means that things like labeling tubes, taking observations diligently are implied. I'm expecting that the researcher is doing that. So you don't necessarily have to include that in your methods section. You also don't have to include kind of procedural actions like opening the bottle of dye. It is important, however, to again emphasize like how much you used. What was the quantity? What did you use? Dye. You combined it with water. So you want to clearly state those important aspects of your approach. Another thing I'd like to emphasize is that these key steps, waiting different time periods and then taking measurements at those time points, is a little bit repetitive. So if you were to write that up, it might feel kind of choppy. So one way to avoid that is to just summarize. So you could say something like, after initial observations, we measure the distance the dye traveled from the roots after 30 minutes and 1 hour. So to conclude this method section, I would suggest to focus on the big picture steps. And so when outlining your method section before you begin writing, focus on, again, identifying what those key steps are, and then creating a flow chart like this. And that can help you get started on your methods. So in this example, you could have the subtitle be this topic sentence, and then again, provide those important details as well. All right, so let's talk about the results. So the major point to emphasize in the results section is to stay objective. So your goal is to share your results, like what did you find? And you're going to describe those patterns and trends, statistical outcomes in writing. And it's important to do that without any form of bias or interpretation. You're also going to show your data as figures and tables. So let's dive into the guidelines. So like the methods, you're going to use the past tense because these events have already occurred. And I'd also like to point out that using to do X, we did Y format and subheaders can be helpful in the results section as well. You're going to focus on describing the trends that you found in your data. So think about providing a high-level summary. You also, again, want to report the results of your statistical analyses. You also want to contextualize. So, I just mentioned you're going to provide figures and tables. Don't start off right away with that information. Provide some of the backstory. What does the reader need to know to interpret that information? It's also important to present your results in the same order as your methods. This can be really helpful for the reader who's trying to follow along and kind of pay attention to what you did and what you found. So let's look at this excerpt from the results section of Lower and Hall 2018. So I'll kind of break down what I notice about how this paragraph is organized, identify their approach, and their findings. So first, their approach is clearly stated here, and you can see that they use the to-do X, we did Y format, and that's how I was able to find that so easily. So their objective was to examine variation at signaling loci, and their approach was to Sanger sequence the 3 signaling loci. And so now I am kind of reminded as the reader what they're about to do. So now we can look at the findings. And so the bulk of the results are really just to describe those findings in detail. So you can see here, these are all the findings. That's most of the paragraph. But I'd like to point out to you how these findings are organized. So if we go back a step, we can see here that these are more general findings. So the authors are starting out by describing kind of broader patterns. So as predicted, all three loci showed sequence variation in coding regions. They also say, however, non-synonymous variation that would result in a change in amino acid sequence was rare or absent. So again, they're showing these kind of broad scale explanations. And so you can see here that they're actually directing the reader to see the data in table 1. So these in-text references are extremely important to include because it helps the reader kind of match up where you state these data in the text and where they should be looking in the rest of your paper. So table 1 includes this information. Now you can see that they transition from these kind of general observations to the nitty-gritty details. So now they're talking about like specific populations, specific exons. They're providing these key details that the reader needs to know. So let's talk about figures and tables. So again, the point of the figure and table is to present your data clearly for the readers. And so you, as the scientist and the author, need to choose how to represent your data clearly and accurately. And one thing to keep in mind is that there are very specific formatting guidelines for figures and tables. And so you'll find them here. And we'll go through these examples, or we'll go through these guidelines using examples. So just be sure when you are creating your figures and working on your drafts that you are following these guidelines. So this is figure 2 from Lower and Hall 2018. And I'll start off with saying there's a lot going on here. They're presenting a lot of data, but the way they're presenting it is very clear. So right off the bat, we can see A and B. They're showing two different types of data. They're looking at haplotype diversity and nucleotide diversity. You can see that they're also looking at 3 different types of genes, which they've recorded as headers. You can also see that they color coded to emphasize that LW is a different gene than UV. They also have reported the axes. So they've been very clear about labeling what the axes represent and then decoding that in their figure caption. I'd also like to point out that the figure always has a caption, which is located below the rest of the figure. So let's talk a little bit more about figure captions. So figure captions are extremely important because, as Dr. Andrea Swigart says, you should be able to look at a figure and get the gist of the experiment. So taking away the rest of the text and the paper, you should be able to look at just that figure and have some idea of what those researchers did. So one way you can do that is by immediately starting off the figure caption with a title, saying what is actually going on in that figure. So provide a concise one-sentence description of what information is actually displayed. And you see that right here. They do a great job of clearly stating what is going on, what data is portrayed in this figure. They're looking at genetic diversity statistics across populations at 3 loci involved in signal production reception. Now, it's also helpful to provide a brief description of the patterns or trends that you really want the reader to notice. So for me, sometimes I'll read papers and I'll just look at a figure and feel overwhelmed. There's so much going on. And so by providing this brief description of a pattern or trend for the reader, you're essentially highlighting it for them. You're telling them what to pay attention to. So that's what they're doing here. They're describing the trend. So on average, adult luciferase had lower genetic diversity than either LW or UV opsin across populations. So they're being very clear about this. Next, you'll see them spending a lot of time decoding these different components of their figure. They're saying what A and B stand for or signify. They're explaining these abbreviations and colors. They're really making it easy for the reader to kind of break down the figure and understand what's going on. So I like to think of figures as illustrations, mainly because it's difficult to interpret data without visualizing it. So for example, Lower and Hall, these authors could have presented their sampling scheme data as a table. They could have just reported the GPS coordinates, whether it was open or closed habitat. All of that could have just been organized into a table. That would be acceptable. But to really make it clear for the reader what they did, they decided to actually plot this map instead. So we can very quickly see, wow, like these populations are pretty spread out. Some are close together. We can see their location proximity to each other. We also get a very clear idea of which habitats were open or closed and what that distribution looks like. So it's important to think about your data and how to present it clearly for the reader. It's also important to remember that the type of data you're using will dictate the plot you make. So for example, if you have quantitative data, which are amounts or measurements, you'll produce a different data or produce a different plot sometimes than a categorical data set, which means that your data is set in groups. So I'd also like to mention that you are all diverse researchers doing these diverse projects. So what you can see here are these different images. And so this author from example paper 2 actually didn't do any plotting for this. They just took these images and then overlaid these circles to emphasize to the reader what they should pay attention to. So you might do that as well if that's the way your data is. So your data will reflect the type of research that you're doing. So for table formatting, like we talked about before, the figure caption usually goes below the figure. So the headers of a table will go on top. So one thing that tables are really useful for are reporting descriptive statistics or numbers really of any kind. So for me, I get overwhelmed reading a lot of numbers in the text. So one thing you can do is just organize them into a table. And that way the reader can just quickly scan across this line and see what information they need to know. I'd also like to mention that, like the figure captions, the table always has this title that is, again, descriptive of the data that it contains. So let's quickly do some practice. Imagine you are a bakery owner and decide to track how many desserts you sell per week. You made this plot so you can compare which desserts are most popular. Your data looks like this. What type of data is it? Well, I would say it's categorical because each of these desserts fits into a group or category. What type of plot could you make? Well, you could make a bar plot, but you could also make a pie chart. So now in one sentence, describe the trend. Well, I noticed that cake, closely followed by cheesecake, are the most popular desserts. Brownies are not quite as popular. How can I improve this plot? Well, say the baker wants to write up a report so that in the future they can look back on this and know what was most popular. Well, in order to do that, they need some cues. So I would have a title for the figure. I would have figure axes labeled, and I would also provide a figure caption so they can really kind of get the gist of what they did and really get all those details and information reported there all at once. Again, with that concise description. All right, so now we're interested in tracking sales since the bakery first opened in 2015. So your data looks like this. What type of data is it? Well, it's quantitative. And not only that, but it's a time course. So that means we're looking at number of cakes sold over time. So in that case, we're going to make this plot that has time always on the x-axis, organized in chronological order. So I could make this line plot, but I could also make a bar plot. So in one sentence, describe this trend. Well, I see a pretty steady increase in the number of cakes sold over the years. All right, so that's all I have. Thank you for watching this video, and I hope to see you for our next video and workshop on writing the discussion and abstract. Hopefully you found this helpful, and if you have any questions or concerns, please contact me and let me know. I'm happy to help. Thanks so much. Have a great night.